Hi everyone. People are still joining in, so we'll give them a few more minutes. All right, I think we might start. We're expecting more participants, but it's already 2 past, so we'll start. Good afternoon, everyone. My name is Tu. I'm head of the Clinical Genomics Lab in the Precision Medicine Research Group at Monash University. I would like to begin by acknowledging the people of the Kulin Nations, who are the traditional owners of the land on which we're gathered today at the MPCCC and across Victoria. I respectfully acknowledge their elders, past, present, and emerging, and I pay my respects to any members of the Aboriginal and Torres Strait Islander community gathered among us today. Together with Professor Melissa Southey, Chair of Precision Medicine at Monash University and Chair of the MPCCC Exec, I would like to welcome you to today's MPCCC Tech Talk. These talks aim to engage the clinical and research communities to share expertise and knowledge about state-of-the-art technological innovations in cancer medicine. We want these talks to be a platform for sharing information and discussion and to enhance multidisciplinary collaborations. So I'm very pleased today to welcome Dr. Bernie Pope. He will talk to us today about challenges and lessons learned from somatic variant calling in the context of whole genome sequencing. He will share his experience from a large international prostate cancer study of more than 1,000 tumor samples, and he'll tell us about VarLab, a software tool he's developed to address the issues of quality control and identification of problematic variant calls. After completing a computer science PhD in programming languages, Bernie undertook an internship at Microsoft Research in Cambridge in the UK before returning to a lecturing position at the University of Melbourne. In 2010, he joined the VLSCI, which is now called Melbourne Bioinformatics. Given his training as a computer scientist, Bernie is particularly interested in addressing algorithmic and computational challenges associated with biomedical research, especially in the context of human genomics and cancer and genomic factors underlying disease risk and progression. In the past 10 years, Bernie has provided his expertise to a number of research projects in breast, colorectal, and prostate cancer. And I certainly know that my research projects would not be where they are today without Bernie. In 2017, he was one of only 3 recipients of the prestigious Victorian Health and Medical Research Fellowship, which really highlights the quality of Bernie's research. Before I hand over to Bernie, I would like to go over a few points of housekeeping for this event. Could all our attendees please ensure you have your video turned off and your microphone muted before we begin? We'll be taking questions at the end of Bernie's presentation. To ask a question, please click on participants at the bottom of your screen and select raise my hand. And please remember to unmute and introduce yourself when asking your question. I'll stop talking now, and I'm very, very pleased to introduce you to Bernie.
Okay, thank you very much, Tu, for that wonderful introduction, and thank you to everyone for coming along to this talk. Okay, so given that this is a tech talk, it does not contain scientific results, so hopefully you weren't expecting those. It's also work in progress, and so obviously we're explaining things that are ongoing at the moment. What I wanted to do, as Tu mentioned, was highlight a couple of solutions that address technical challenges in somatic whole genome sequencing, These solutions arise from the Pan Prostate Cancer Group and also the Portable Pipelines project, and I'll be talking about those two projects today. Okay, so first of all, in part one, the Pan Prostate Cancer Group. So the Pan Prostate Cancer Group, or PPCG, is a large international consortium of researchers investigating prostate cancer around the world. Currently, we have accumulated 1,200 tumor samples approximately, with matched blood germline normals, and also detailed pathology and clinical data with all of the samples. We're also collecting RNA-seq and methylation data, particularly 450K methylation data for an overlapping subset of the cohort. The overall project is modeled on the ICGC Pan Cancer Analysis for Whole Genomes projects. It's often called PCORG, which recently had a significant amount of publications released. And there's the logo for the PPCG, and here this map of the world with stars on it, you can see the positions in the world, the geographic positions where the collaborators and data are from, and down at the bottom is a link to the website for the project. So one of the key things about the bioinformatics in the PPCG is that all of the primary bioinformatic analysis, which is alignment of the sequences to the reference genome and basic variant calling and some other analyses, have been done in a distributed fashion at each of the contributing sites. Obviously, 1,200 approximately tumor genomes is a lot of analysis to do. And one of the key things about that analysis is that we have used containerized pipelines, which execute identical analysis. So they run exactly the same software on all of the samples, and that makes sure that we're doing a consistent analysis over the entire data set, which is very important to avoid batch effects and other things associated with if we ran different pipelines, we could potentially get different results just by virtue of using different tools. So we're using exactly the same tools and analysis pipelines, and that has been done by using a technology called Docker containers. And so even though the analysis is distributed, it's identical. And the two pipelines that we are using are derived and updated from the PCORG. They are pipelines from the Sanger Institute and the DKFZ. Apart from calling mutations in the tumors, we are also performing the following primary analyses in the PPCG, and then these will feed into downstream analyses in specialist research groups. And in this table, I have highlighted most, if not all, of the primary analyses that we're doing in the PPCG. I just wanted to highlight the kinds of things that you can do with whole genome sequencing in a somatic variant calling context in cancer. And on the left-hand side, I've identified the analyses where I think whole genome sequencing is critical, so that we wouldn't be able to do these analyses without whole genome sequencing. And in the right-hand side column, I've identified analyses where I think whole genome sequencing is highly beneficial, though you could probably do some of those using targeted sequencing technologies such as exome sequencing or capture in some circumstances. So in the critical column, these analyses include detection of focal hypermutation events. These include something called contagious, and I'll do a little case study in contagious later in this talk, and chromothripsis, which is a focal hypermutation event related to structural variation and catastrophic genome fragmentation. We also analyze structural variation and chromoplexy. With structural variation, the reason you need whole genome sequencing is because the break-ends, the DNA break-ends that occur as part of structural variants can lie anywhere in the genome. And if you have a targeted sequencing assay, then you may miss those break-ends. So it's very important to have the whole genome to detect the full array of structural variation. Chromoplexy is another kind of complex structural variation event that is similar to chromothripsis, though tends to involve fewer mutation events and also an increased number of chromosomes. We are also investigating clustering and evolutionary reconstruction of subclones within the tumors. So this is looking at the clustering, particularly single nucleotide variants, although this can include other types of variants as well. So this is clustering the variants based on their cancer cell fraction and also relating them to the copy number variation that you see, so that you can group variants based on the estimated subclone of cells that they derive from in the tumor. And then we can look at the evolutionary history of those, and we can order the events of mutations. And in cases where we have primary tumors and metastases, we can also determine from which clone the metastatic sample has seeded from. We're also interested in timing of mutational events, whereby you can take the clock signature of certain mutations that happen over time, and we can use those to order temporarily the mutation events that we see. For example, we could look at the ordering of subclones relative to time as well. And in order to calculate the timing of mutational events, we need a very large number of mutations, particularly because we're interested in gathering as many of these clock mutations as possible. And so we need to have a very large collection of mutations. And so that's pretty critical to have whole genome sequencing there, particularly in a cancer such as prostate cancer, which is typically known to have a lower mutation count than some other cancers. We can also look at the microbiome in the cancers by looking at reads that don't map to the human genome reference, or that are collections of DNA sequences or fragments that have arrived from viruses and bacteria that might have infiltrated the tumors. And we can look at telomere length as well, and shortening of telomere length is known to be related to disease progression. In the right-hand column, where we've got whole genome sequencing is beneficial, like I said, you could potentially calculate, and people do calculate these using targeted sequencing assays, and we're considering somatic mutation signatures that have been famously published on the COSMIC website. We're looking at driver mutations. Again, having whole genome sequencing is beneficial for calculating driver mutations, because you need to determine driver mutations, you need to compare mutation rates in genes compared to background mutation frequencies in the local genomic context. And therefore, having the context around the genes is rather important for doing that. We're looking at copy number aberrations, including whole genome doubling. You can estimate copy number aberrations using other assays, although correctly ascertaining the break ends of the copy number, just like structural variants, generally requires whole genome sequencing. Another thing that's quite interesting that you can do with whole genome sequencing, and it's sort of a new idea to me, is that you can use your tumor sequencing to phase germline variants. So actually, if you have a tumor sample and a germline sample, and the tumor sample has regions of imbalanced copy number, you can use those regions of imbalanced copy number to determine the phasing of germline variants that you can detect in the tumor, which is actually quite an interesting application of somatic mutation sequencing, where you're actually using the tumor to inform information about the germline. There are many challenges in accurate somatic variant calling, and to name a few of the technical challenges we encounter, typically we have some samples with low tumor purity. So in pan-prostate cancer group, like most studies, we have a range of tumor purities across the cohort, and we can deal with tumor samples with low purity, although that tends to affect our sensitivity for calling variants, because we have an increased amount of non-tumor sample in the sequencing. Tumors tend to be heterogenic and also tend to have subclone populations. So then when you're doing bulk sequencing, as we are here in most studies, you're sampling from a population of cells. And then because of this heterogeneity, you may not see a consistent signal across all of those cells, and that you may actually want to detect infrequent mutations from the population of cells that you've sampled. So you need to be able to detect variants with low variant allele fraction. We can have in cancer highly disrupted genomes, which tends to complicate the detection of variation, and also just complicates the alignment of sequences to the genome. We can have examples where we have low depth of sequencing, even if the mean depth of sequencing across the tumors is quite good, just due to the normal variation that you'll get through sampling. You will end up with regions with lower sequencing coverage than you might like. And again, that affects our ability to accurately call variants in those regions. And we also have, particularly because of the subclonal populations in the cancer cells, and particularly because of low tumor purity and tumor heterogeneity, we end up with the low variant allele fractions in our samples. On the right-hand side, here's just a diagram of a particular region in a couple of tumor samples, just identifying how messy the alignments can actually be. And anyone who's looked at tumor genomes might have seen things like this before. And these are regions where we have a vast number of changes compared to the reference genome. And this is likely to be caused by contamination in the sample. And so you wouldn't want to actually take whatever changes are happening here and believe they are actually related to the true biology of the tumor. So that's leading me to a problem related to quality control. And the key point is that we need to separate the signal from the noise. And that's a basic problem that we're facing with bioinformatics. At the very beginning of the analysis where we're doing variant calling, and this is especially true at low variant allele fractions, where the true variants are mixed with a collection of sampling and sequencing artifacts and effectively noise. And so we have faced this challenge that we want to actually detect variants at low variant allele fraction, but we also need to avoid polluting our data with noise and error. So one approach that people use to solving this problem is to do consensus calling using multiple sequencing pipelines. This is some data from the PCORG. In the PCORG, they took an approach where they applied 4 different variant calling pipelines, and they retained any variants that were called in at least 2 out of the 4. This is called a consensus calling method. This is pretty common technique, and people apply this technique because they're trying to balance sensitivity and specificity. If you were to just take the agreement between all of your callers, then obviously you would reduce your sensitivity because you would be removing true variants that happen to be missed by one or more of the callers. If you took all of the callers, then maybe you would be reducing your specificity by increasing the amount of noise in your results. And the plot on the left is something I have created myself from 8 of the tumor samples that we used in validation for the PCORG. And this is just showing you on a log scale on the vertical axis, the number of variants that were called by the intersection of various calling pipelines that are named Broad, DKFZ, Muse, and Sanger. Most of the variants that you see in the data are actually called by all of the callers. But you can see that there's a fairly large proportion of variants in the data set that are actually not called by all of the callers. And so inevitably we're facing this challenge of deciding which variants should we keep and which variants should we remove. In the PPCG, we've adopted a slightly different approach to filtering our variants. We call variants, as I mentioned, using 2 PCORG derived pipelines, the Sanger and the DKFZ pipelines. We've discovered through our analysis that the best trade-off between sensitivity and specificity has been achieved by doing the following things. First of all, we take the union of the pass variants that are generated by each pipeline, so from those 2 pipelines. So the pass variants are just the ones that the underlying variant caller thinks are acceptable for further analysis. We remove variants that were occurring in regions known to be problematic for alignment, and for example, the ENCODE blacklist. And I should mention we're still using HG19 reference genome for historical reasons. We also, and probably this is the key thing that we're doing that makes the biggest difference, is we remove variants with supporting evidence from a panel of normals derived from the Sanger Institute of roughly 100 germline samples, where the variant calling pipelines are applied to germline samples. And then we look for evidence of somatic mutation calls in the germline. And those are indications of either germline variants that have mistakenly been called in the tumor, or more likely those are artifactual regions where, because of alignment artifacts and other problems, the variant callers have called false positive variants. So this plot here shows the effect of that filtering technique on 898 tumors. You might notice in my slides today that I have varying numbers of tumors in each of the slides. That is a consequence of different analyses being done at different times with different amounts of the data set being available. On the left-hand side, we see a plot showing the unfiltered variants that are just the pass variants generated by the Sanger and the DKFZ pipeline, which is called EO in this for early onset. It's a historical naming change, so it's called the EO pipeline in this plot. That's the DKFZ. And in the right-hand side, we see the final filtered sets. Each circle is a sample from the study, a tumor sample, and they're colored by the tissue of origin. So we have primary metastases and normal benign prostate tissue. The vertical axis shows the percentage, the proportion of variants, I should say, per sample that are in the consensus between the two variant callers. So as you travel upwards in these plots, you are seeing samples with higher proportion of variants that were called by both pipelines. As you travel down in the plot, you're seeing samples with a lower proportion of their variants called by both pipelines. And then the axes on the horizontal plane show the proportion of variants in the sample called by the EO pipeline, which is the DKFZ, and the proportion of variants called by the Sanger pipeline. So if you travel down, if you look at the left-hand side plot and you travel down to the right-hand side corner there, you will see a collection of samples, tumor samples, that have a very large proportion of DKFZ or EO variant calls. So they're predominantly, the variants in those samples were called by one pipeline only, and that's in the unfiltered set. After doing the filtering that I described before, you can see that all of those samples have shifted away from that corner. And so for some reason or other, in those particular tumor samples, particularly normal benign prostate tissue, the EO DKFZ pipeline is very sensitive to changes in those samples, which we've removed by doing the panel of normal filtering. You can also see that the circles become smaller. Each circle is proportional to the number of variants that were called in the actual sample itself. So basically by filtering, we're removing quite a large number of variants in samples. Overall, what tends to happen is that the individual samples shift towards the consensus corner of this plot. So basically by filtering, we're tending to enrich for variants where both of the pipelines have called the majority of variants in their samples. Although you do see some samples which are still dominated by variant calls from the Sanger pipeline in the bottom left-hand corner, which is an interesting phenomenon. We generally know that the Sanger pipeline is a bit more sensitive than the DKFZ. Despite every attempt to filter variant calls correctly, errors will persist in our data. This is a natural consequence of the trade-off between sensitivity and specificity. There's sort of nothing we can do about this, that we're going to end up with errors in our data. And so particularly for some analyses that we want to do, we want to err on the side of sensitivity, particularly, for example, subclonal reconstruction, where we need to actually detect variants of low variant allele fraction in our data because some of the subclones occur in a very few number of cells that we were sampled when we took a tissue sample. So this means that we will have false positive variants in the final data set. And the question is what to do about those. So that leads us to VARLAP, which is what Tu mentioned at the beginning of the talk. So we recognize this problem and we've developed a tool to partially address this issue. And VARLAP automates the process that many bioinformaticians would do manually when encountering variants where they want to assess the quality of the variant. And basically what VARLAP does is it considers each variant in its alignment context, which is in the alignment file in BAM format. And it looks for evidence for potential problems, for example, poor quality of alignment. It reports a large table of statistics about each variant and what it finds in the alignment context of those variants. And this can be used for QC and further filtering. Here's a diagram to show you roughly how it works in principle. So one of the inputs is a VCF file, which contains your variants of interest. Most of the time, these will vary in the context of somatic variant calling. These will be variants that are derived from a particular tumor sample. And then you supply one or more BAM files, which are the alignments of the raw sequencing reads to the reference genome. In the context of somatic variant calling, that would typically be a tumor alignment and a germline alignment. And so for every variant in the input VCF file, you get a row in the output table. It contains all of the usual information about the variant, including its position in the genome. It contains some information, read alignment independent statistics that are not depending on the actual reads themselves, but some interesting things about the variant, that might be relevant to its locus in the reference genome. And then for every BAM file that you've provided on the command line as input, it produces a collection of statistics about that variant in that BAM file. And these are some examples. For example, it contains statistics about all reads that overlap the variant locus, so regardless of the actual DNA in the read. It also contains statistics about reads that support the reference allele, and it also reports statistics about reads supporting just the alternative allele, so the alternative allele that was recorded in this variant. So that gives us a way to see what the overall picture is, what's going on with the reference reads containing the reference sequence, and also what's going on with reads containing the alternative allele sequence. These are some example metrics, so it computes many more than these. They're mostly related to the alignment quality, so base quality of the DNA base at the variant position, mapping quality of overlapping reads, number of mismatches in overlapping reads, which is called NM in BAM file nomenclature, and it stands for edit distance, the alignment length of overlapping reads, the counts of each DNA base at the locus, number of indels in overlapping reads, number of clipped bases, number of overlapping reads on forward and reverse strands. And you can also supply other information in a bed file, and it will tell you whether your variant overlaps a genomic region of interest, such as a repeat region, for example. So what I wanted to do is show you a case study based on Catejus, where we've applied VialApp to good effect. So this plot here is called a rainfall plot, and it's for a single tumor sample on chromosome 2. Each of the dots is a single nucleotide variant that was detected by our variant calling pipeline, and these are filtered using the panel of normal filtering approach that I mentioned previously. The horizontal axis is the locus on the chromosome in base pairs, and the vertical axis is something called the intermutation distance, which is the distance between adjacent single nucleotide variants in the data set on a log scale. And then each variant is colored by its base change. And I would like to thank Brion Ferran from Tony Pappenfuss's lab at WeHi for generating this data and this plot. So the key thing with Catejus is its focal hypermutation. It's technically commonly defined as 6 or more SNVs with an average intermutation distance of less than 1 kilobase. When you plot it in this rainfall plot with a log scale of the intermutation distance, when you see Catejus, you see these stripes of very close mutations. So these are mutations that are all close together, therefore their intermutation distance is low. And it's called a rainfall plot because it looks a bit like rain dripping down a window. And you can see that in this particular example that the mutation changes are predominantly C to T and C to G. And C to T and C to G changes are also a characteristic of Catejus that's related to mutations caused by the Apperbeck enzymes or translational DNA synthesis polymerase. So if we cluster this data based on the base changes, so this data is clustered on base changes, each column in this plot is an individual categic region that's been detected over 867 tumors. We can see that we get overall reasonably good clustering of the categic regions based on the base change. Predominantly C to T and C to G changes are determining this rightmost cluster here. On the left-hand side cluster, we have quite a mixture of base changes. Additionally, this is a plot generated by Brion as well. We have single base substitution mutation signature 13 and signature number 2, which are also indicative of the kind of mutation patterns caused by Catejus. And the colors here indicate the proportion of the mutation signature in that categic region associated with those signatures. And so we see an enrichment of SBS 13 and SBS 2 in the right-hand cluster. We also have a indication of how many donor samples contributed a categic region that overlaps this position. What we hope to see, generally what we expect to see, I should say, is that the categic region should be roughly random in the genome. And generally, we do not expect them to overlap across different donor samples. That is, tumors from the same individual, we expect to have unique categic regions in our overall cohort. What we do see here, though, is that on the left-hand side, we do see a lot of overlapping regions, which is a sign of something going wrong there. But also we do have a smattering of overlapping regions on the right-hand cluster as well. This dot plot at the bottom here shows the size of the categic region in log 10 base pairs. We generally expect the categic regions to be larger regions of the genome, and so we expect that true categic regions should have higher dots. The dots should be higher up in this plot because they should be longer, hundreds or thousands of base pairs long. And finally, in this plot, we have strand bias, which is indicating a proportion of the reads in the categic region being biased to one or one of the strands, the DNA strands. We know from the literature that categus is associated with DNA damage to a single DNA strand. Again, we see an enrichment of highly strand biased mutations on the right-hand side, but we still have a mixture of some categic regions with low strand bias. And so what we want to do in this data is actually separate the true and false categic regions. We put those features that from that previous plot into a PCA, and we've noticed very good separation between them. In particular, the pinkish orangey dots on the left-hand side of this principal component plot, we believe are high quality categic regions. They're enriched for C to G and C to T changes. They're also enriched for strand bias and for the size of the categic region. And we can see that the first principal component can explain about 40% of the variation in its data. So they separate very well. On the right-hand side, we have other features that we do not that are enriched and we do not expect to be related to categus. So we applied k-means clustering where k is 2. So we basically clustered these into 2 groups and assigned the left-hand side group to a high quality group and the right-hand side to a low quality group. After doing that, assigning to groups, we've put the high quality categic regions on the left-hand side here and the low quality regions on the right-hand side. And you can see that this has really separated the two quite nicely. In particular, the high quality categic regions, except for maybe 1 or 2, are no longer overlapping across tumors from different donors. So that raised the question of, well, what's going on in this low quality categic region data set? Is this something else that's interesting in the genome, or is it just noise? And so we applied VARLAP to those 2 groups of variants and compared the results. This plot here, I should just mention, is indicating that the high quality variants are enriched for C to G and C to T changes in trinucleotide contexts that we do expect to see for categic regions, whereas the low quality ones are not. So after applying VARLAP to all of the variants that fall into those categic regions and classifying the variants based on whether they're high quality categic regions or low quality, we can compare the statistics that VARLAP computes based on the read alignments to see whether we think there's an issue with any of the data set. And so on the left-hand side, this distribution plot shows the vertical axis is just one particular metric that VARLAP produces out of many that it considers. This one happens to be quite informative in general, which is the average edit distance of the sequencing reads against the reference genome. And this is considering just reads that contain the reference allele. And you can see there's a little bit of a difference where the low quality categic regions are enriched for a little bit of extra edit distance. So they're not mapping as well to the reference genome as the high quality variants are, but they're not too different. However, we see a striking difference on the right-hand side when we look at reads that support the alternative allele. In reads that support the alternative allele in the tumor, we see a massive difference between the edit distance in the low quality variants from the high quality variants. This is telling us that it's most likely the case that the low quality categic region variants are actually derived from mapping sequence artifacts, not true biology. If we take all of the data that's produced by VARLAP and then we put it into a random forest classifier, we can see that, and we try to classify whether the variants are from low quality or high quality regions, we can see that we get exceptionally good separation between the two. Under cross-validation, we get nearly 97% accuracy in classifying every variant to whether it's from a high quality or low quality group. And this plot on the right-hand side here is just telling us the features that are most important in the classification. And the most important one is the average edit distance of reads that support the alternative allele. Again, just indicating that there's something difficult with these regions where we're having trouble aligning the reads correctly to the reference genome. And that's indicated by the fact that the reads are quite different from the reference genome, indicated by the edit distance. Okay, just quickly, a thing I wanted to mention is that another thing you can do with VialApp is something I'm calling cohort analysis, where instead of taking variants from a particular tumor sample and then checking them in BAM files or alignment files for that sample, you can instead take all of the somatic variants in your cohort, and you can compare them across all of the BAM files in your cohort. And then you can compute something called cohort VAF, which is just adding up all of the alternative alleles across your cohort for every variant against the evidence that the depth of sequencing. So this gives you a variant allele fraction for every variant over all of your samples. You could look in the tumors, or you could look in the normal or the germline samples as well. And what you can do, what you can use this for is you can find variants that have an unexpectedly high variant allele fraction across your cohort, which is indicative again of some kind of problem occurring at that locus. This plot here is log scale on the vertical axis, and it's showing a histogram of the cohort VAF across 66 tumors that I happen to analyze in this particular data set. Most of the variants have a very low variant allele fraction in the cohort. That means that basically the somatic variants we're detecting are actually not occurring in any evidence across the rest of the group. Although you can see that there's tens, if not hundreds of thousands of variants on the right-hand side of this plot that do occur at an unexpectedly high VAF in the cohort. That is, there are sequencing evidence for those somatic variants across the cohort at an unexpectedly high rate. And so what you can do with this kind of analysis with VialApp is you can actually use it as a kind of filter as well, where you can remove variants that occur in your data set at too high a frequency to be expected to be real, because most of the time we expect it to be relatively rare. Okay, so VialApp is open source software. You can access it from GitHub there, and there's documentation about how to use it. It is independent of the underlying organism, sequencing assay, and variant source, so it makes no assumptions about how you're going to use it. I've mentioned how you can use it in a couple of ways for somatic mutations, but it's equally usable for germline variant analysis if that is something you might be doing. Also, it could work on different organisms. It currently supports SNVs and short indels, and work is underway with a student to also support structural variants as well. Okay, so I'm going to move on quickly to part 2 of my talk, which discusses something completely different. It's the Portable Pipelines project. This is, I should mention, not part of the pan-prostate cancer group. It's a separate project. But I have talked a bit about variant calling pipelines in the previous part of the talk, and many people here will be interested in running variant calling pipelines on their data. You might already know this, but these variant calling pipelines are extremely complex software systems that are composed of dozens of components, particularly separate tools for doing different tasks. For example, different variant calling tools, different quality control tools, and so on. These pipelines are difficult to write and maintain, particularly for research groups that don't have a lot of resources to put towards these things. They are hard to validate. So even if you have a variant calling pipeline, how do you know if it's doing a good job? They're often highly dependent on the computing environment in which you run them. For example, they might run on the cloud or HPC or a local server, but maybe not across all of those things. And these days, many research groups have access to a variety of computing resources. And therefore, there's a lot of demand for being able to do your analysis in different environments. Also, one problem we've noticed and why we started this project is we have lots of repeated effort across research groups to develop essentially the pipelines doing the same thing, but we do it in different ways, and we are trying to avoid that now. So the Portable Pipelines project is a collaboration between the Peter MacCallum Cancer Centre, Melbourne Bioinformatics at the University of Melbourne, the Walter and Eliza Hall Institute, and the Australian Biocommons. The project aims to build bioinformatics pipeline infrastructure that is portable across institutions and environments. And we want to enable greater sharing and reusability of these pipelines. We don't want to be each repeating redundant work. We want to share our resources where possible and get on to the more interesting things in our research. So we have lofty goals. We want our pipelines to be portable. We want them to be standards compliant. We want them to be modular so that you can mix and match pipelines and switch features in and out. We want them to be extensible so that you can make updates to them. And we want them to be easy to use. And so that's our ambition. We've created a tool called Genus, which is a software realization of these ambitions, which I'm going to talk briefly about just now. So you might already know that there are many workflow standards out there. There are literally hundreds of bioinformatics pipelines, even those related to cancer. There's hundreds of those. And there are some standards emerging, particularly the Common Workflow Language is one. There's the Workflow Description Language, some people call it WDL or WDL, and there's Nextflow, which is also a quite popular standard for developing pipelines. And as the saying goes, the great thing about standards is there's so many to choose from. And so the question is, well, what standard are you going to go with? And the risk is that you pick a standard and then it disappears in a few years' time and you're back to square one, making new pipelines again. So recognizing this problem with multiple competing standards, we have created Genus, which is basically a small layer on top of the standards. Basically, you write your pipeline in Genus notation using Python, and then that is converted to any one of the standard pipeline formats. It currently supports CWL and WDL, and there's work underway to support Nextflow. The benefit of doing things this way is that we leverage all of the resources that those standards have to be able to run pipelines under different execution engines, and there are many of those, and those engines support all the different computing environments that I mentioned. And so Genus basically consists of two components. You write a pipeline specification in Python in a very high level way. That can be translated into CWL, WDL, or some other standard notation. We also have an assistant called Genus Assistant, which helps you run the pipeline under some environment of your choice. And so you have both a way of translating a standard specification into, a specification in Genus into a standard, and you have a way of running that on the computer and orchestrating all of that work for you. Genus comes with pre-built pipelines. So if you're not interested in making new pipelines, you want to just run existing standard pipelines that follow best practices, we have those already built. We have, for example, a whole genome sequencing germline pipeline based on multiple variant callers. And we have a somatic sequencing pipeline based on multiple variant callers as well. We have quite a number of other standard pipelines that you can use that are pre-built, ready to go, and straightforward to run. So if you have some data that you would like to analyze in a fairly standard way, then Genus may be something that will be quite interesting to you. Genus availability, it's ready to go right now. We have quite extensive documentation online. So you can find the Genus documentation by searching for Genus pipelines in Google or following these links. Getting up and running in Genus is relatively straightforward if you're familiar with the command line. It's just a few commands and you're ready to go and execute a pipeline. And we have the source code maintained under GitHub as well. We are very interested in collaborating with research groups. We would like people to use Genus as much as possible. And so if you're interested in running variant calling pipelines on data and developing pipelines or using existing ones or developing new ones, then we would love to talk to people about that. And we are running workshops regularly about getting up and started and using Genus. So that ends my talk just there, just in time. And I would like to thank many people for contributing to this work. Many people from the Pan-Prostate Cancer Group. I haven't mentioned everyone. I would have to have hundreds of people on this slide if I mentioned everyone, but specifically for the work I've mentioned here, I would like to thank colleagues from the University of Melbourne, from WeHi, the University of Manchester, University of Copenhagen, and OICR in Canada. And for the Portable Pipelines project, I would like to thank colleagues from the University of Melbourne, WeHi, and Peter Mac. And that ends the slides for my talk, and I'm happy to take questions if there are any. Thank you.
Thank you very much, Bernie. That was a fantastic talk. I'm sure we have questions Please unmute yourself and yes, Nick.
Hi Bernie, how you going? Great talk.
Thanks, Nick.
I've just got if it's okay, I've got two questions. One of them is, you're using whole genome sequencing data. Is your kind of pipeline compatible with deep amplicon sequencing or amplicon panels?
So the pipelines I think you're mentioning are from the Portable Pipelines project with Janice. We don't have a specific pipeline for amplicon sequencing at the moment, though.
No, sorry. I was just more thinking of the observations just in terms of semantic variant calling.
Oh yeah.
And the, I suppose, the filtering logic that you're using behind the scenes.
Oh yeah.
I'm just wondering if those kind of quality metrics and things are actually compatible with a much deeper per base coverage.
Yep. Oh, I see what you're saying. Yep.
Yeah.
Yeah, so that's a great question. So VileApp, that tool should work with deep targeted sequencing for sure. And like I said, it's agnostic to the underlying technology that you're using. It could be quite, in fact, we have used it for targeted sequencing in a different project completely that I haven't talked about today, detecting circulating tumor DNA, where you want to do very deep sequencing as well. And again, the problem in that kind of project is you want to detect variants with very low variant allele fraction. And so you want to be able to separate those variants like we did with the strategic regions from, the noisy ones from the true ones. And so, yeah, definitely VileApp would be suitable there. In terms of cohort filtering, again, I think that would work very well. Cohort filtering is effectively an extension or an elaboration of the idea of using a panel of normals. And so, yes, if you have it, I guess the kind of summary of everything I've been saying in this talk today is one of the things that's become apparent to me about whole genome sequencing is its power of numbers. So that having lots of data available, which is what you get with whole genome sequencing, lots of data is very powerful for determining events which are not on the per variant basis, but on the aggregation of variants. With targeted sequencing, I guess you've got a lot of data in the other dimension, which you've got very large amount of sequencing depth.
Thanks. My second question was in relation to your sensitivity specificity, again, from the first part of your talk.
Yeah.
Did you have a look at how many false negatives that came out of that? So things that, are true variants, but have been thrown out.
Yeah, that is a lot harder to assess. So false negative, yeah, you're right. I mean, I focused on false positives in this talk. False negatives are hard to assess. What we have done with the PPCG is we've used the PCORG validation data set. And so the PCORG has sequenced 50 tumors and resequenced variants at very high depth. You can estimate some false positive calls using that kind of data, but the problem with detecting false negatives, I should say, the problem with detecting false negatives is you don't have such a good resource on the ground truth, right? Because you're missing things, so how do you know what you're missing? But if anyone has great ideas about dealing with those, I'd be happy to discuss.
Thanks, Benny.
Thanks, Nick.
We have time for more questions.
Hello.
I have a question, if I can be so bold.
Lavinia, please.
Thank you. Thanks, Benny. That was a really interesting talk. So as you mentioned, 1,000 genomes is a lot of data. So I'm interested in really two aspects of this. Firstly, I'm assuming the consortium put together a list of criteria at the beginning of the project. Did they have limitations on tumor purity, on how the sample was obtained, and the sequencing protocol? So did all the samples undergo the same sequencing method? And my second question is, now you've got all this data, what are you going to do with it? Has it got consent? That how is it going to be available? Are you going to have an access committee? Do you intend to put it in something like EGA or more open data repository? What are your plans?
Yep, all those are excellent questions, Lavinia. So the first question was, were they all sequenced in the same way? They were not sequenced in the same way because this is an accumulation of data from different research groups. So the goal is to get as many tumors as we can together because we want power of statistics for those. I agree that sequencing them on the same platform and everything would be brilliant. That's not feasible for this project. However, the bioinformatics pipeline is the same thing as I mentioned, so they're identical analysis for the pipeline. I'm trying to remember the other part of your question there. So in terms of data availability, so the project is ongoing, but the data will be available on the EGA eventually once all of the research is done. There will be a standard data access committee approach to accessing the tumors and so on. So and in terms of tumor purity and selection of samples, I'm not 100% aware of whether there were any criteria for that side of the project, although we have a quality control process I haven't mentioned in this talk where we actually go through all of the samples and we grey or blacklist samples based on other criteria, for example, low tumor purity, low depth of sequencing coverage, many other factors that I don't have time to mention that are basically removing or indicating samples that may be difficult to analyze in certain ways. What we intend to do with all of the data, I think you asked, well, there's a lot of things we intend to do. I mentioned some of the primary analyses that we're doing. There are many downstream analysis groups that are looking, for example, I'll just mention a couple is tumor evolution. So we have primaries and metastatic samples. So how do the primaries evolve to metastatic disease? Looking at disease progression, we're looking at associations with clinical outcomes. And many more things. And I guess one of the key things I did mention in the data is that each of the samples is associated with detailed pathology and clinical data, including recent follow-up with the patients.
Okay, well, that is interesting. Was there any consensus on how that had to be collected, or are you again just taking what's available?
No, okay, so there is a data, there's an entire working group for the pathology and an entire working group for the clinical data curation and collection. That's an ongoing process. We've established data dictionaries for those, which are based on the standard data dictionaries you might see in other studies, tailored for the project that we're working on. There's a complete review of the pathology as well, so re-review of the pathology samples as well, and review of the clinical data and standardization of nomenclature and measurements and so on.
Very nice. I'll be fascinated to see that when it's made available.
Yeah, great. Thank you.
Thank you.
I'm going to ask a question from the chat. In the meantime, please feel free to raise your hand so you can ask your question just after. So I was asking, does the pipeline have any tools for non-coding variants, especially for non-coding drivers predictions in cancer?
Depends which pipeline is being referred to. So for the PPCG, we're using the Sanger and DKFZ pipelines. Those pipelines more or less produce variant calls. There are downstream tools that do the driver detection, for example, which include the drivers that were mentioned in the question. So non-coding drivers, all sorts of different, including single nucleotide variants, structural variants, copy number, those things all together. For the portable pipelines project, the answer is no. There's no specific support. The portable pipelines project at this stage has established pipelines for variant calling. If you want to do downstream analyses, then those are not incorporated at the moment. I guess the simple answer for that is if people would like to work with the portable pipelines project and establish analyses, then we would love to talk to people about what they want to do. And I guess it's driven by demand to some extent. What's a useful thing for people to have?
Okay.
Hi there, Benny. Can I ask a question? Yeah.
Yep.
So I was trying to figure out how to raise my hand on Zoom. Oh, here you go. Raise my hand. I got it done. The thing about the edit distance that you mentioned, and the fact that it really separates the two, I guess phenotypes, whatever they're called. That's a really awesome bit of knowledge to know that there's obviously the reads that map really badly, and those that you want to throw away. And this probably applies for other things as well. And you've mentioned structural variants. I had a quick pick at one of my fusion calling StarAligner BAM files. They don't seem to have an M flag, and I guess that's because they have to have this split kind of, have to split their read in multiple ways, right? Is that, so how do you, so I think you mentioned you're developing, is that you're just basing things on Cigar? Is that how you're trying to get around things? And
Yep. So you asked a question about whether you could apply it to transcriptomics data, I think. Yeah. So I haven't applied VALAP to transcriptomics. It's a good question. Obviously, with transcripts, you have that issue that you mentioned, that the actual sequencing reads get split to the exons. And so that would be challenging for the way that VALAP works at the moment, in the sense that it might report statistics that are not relevant for the quality there. I think it could be adjusted for transcriptomics, although I haven't put any effort into that at the moment. Edit distance should be something I think that the read aligner for transcriptomics should still report. I haven't looked to check, but I expect it to be there. Having said that, VALAP produces many dozens of metrics for every variant, and it's actually growing in what it computes, and most of it's derived from the alignment information per read, but there is some aggregate information as well. But what I wanted to say was that it's not just edit distance that turns out to be important. It is one of the more important things, for example, in that strategic analysis, but many other metrics that VALAP produces potentially could also be useful in quality control for transcriptomics. It's just not something I've looked at the moment. If you wanted to try it out and apply it, then I'd be happy to talk about it further.
Yeah, right. Then that's awesome. Thank you. I guess I was just thinking a little bit more in a slightly different direction, where I was thinking that in general, whatever analysis that we do, those reads that badly mapped with the high edit distance, in general, whatever we do, we kind of want to throw them away or flag them or something like that, because I guess particularly, with detecting variances or structural variances, they could mess a little bit with the workflow. But anyway, thanks for that.
Yeah, well, it made me think of something I wanted to say, which is what VALAP is essentially doing is allowing you to peek inside the black box, which is a variant caller. So right, a variant caller basically, you might think it's strange. Well, hang on, wait a second. The variant caller uses some of this information for determining whether it thinks a variant is true or not. So how come you're able to get more information by going back and looking again? And I guess the thing that's challenging from the point of view of bioinformatics is you're not sure when you get a variant call why a variant call was made or not. And basically VALAP is an attempt to kind of essentially, like I said, peek inside that black box, maybe look at some extra features that the variant caller has not considered, and because it's looking a bit more further afield than just the locus where the variant is. And so that contextual information seemed to be quite important. What you could do is feed that back into a variant caller and have a more sophisticated variant caller that was actually able to accommodate that information as well.
For sure. Yeah, no, that's awesome. Thanks, Benny.
Thank you.
Hi, Bernie. This is Suresh. May I ask a very quick question?
Hi, Suresh. Yes, please.
Hi, Bernie. I was just curious about the Sanger and DKFZ pipelines, and what do they use as a pipeline framework?
Sorry, I missed the end of that. What do they do?
What do they use as a pipeline framework?
Oh, right. What does the Sanger and the DKFZ use? They are bespoke software systems that they have written them. So one of them uses CWL somewhat inside the Sanger pipeline. DKFZ is essentially built out of bespoke scripts that are some written in shell scripts and other languages. In some sense, as far as the PPCG project goes, we are not leveraging some technology for running the pipelines. We're essentially putting them all into a Docker container and running them as a closed system. The benefit is that they're identical across the computing centers. The downside is that it's, again, it's an abstract thing, and we're not able to use something like the Portable Pipelines project to run those pipelines. So that's a trade-off. It's the kind of trade-off that you might make in a large collaboration where you're running pipelines around the world in different centers. But yeah, unfortunately, they're not built on something that was reusable outside of those pipelines.
So if it's a Sanger pipeline and that was using CWL, so inside the container, you are essentially using, for example, a CWL engine to run those pipelines?
Yeah, they use CWL a little bit, but there's other bespoke software they've written to manage the pipeline itself.
Okay, cool. Thanks, Bernie.
Thank you, Suresh.
All right. it's unfortunately the end of this Tech Talk. Thank you very much, Bernie, for your presentation. And thank you everyone for your questions. This was the last Tech Talk for this year. They will resume again in 2021. Sign up to the MPCCC newsletter to what our next talks are. And if there's a particular topic you'd be interested in hearing about, please let us know. Right, so thank you everyone. Have a great afternoon and a very good weekend.
Thank you, bye-bye.